Welcome to today's zoology class. So we have seen in the last class the experiments conducted by Thomas Hunt Morgan on Drosophila melanogaster or fruit fly. So he conducted dihybrid crosses on the fruit fly and he arrived at certain conclusions that we have seen in the last class. So we have to study two crosses conducted by T.H. Morgan on Drosophila. Cross A and cross B. So in the last class we have seen the cross A. In that dihybrid cross, he used two characters. One is the body color and eye color. So as far as body color is concerned, two traits are there: yellow body color and brown body color. As far as eye color is concerned, white eye colored individuals are there and red eye colored individuals are there. And as a result of this dihybrid cross, he got the F2, F1 and then F2. And the F2 he examined the percentage of parental combination and recombination. And parental combination he got a high parental combination percentage, that is 98.7%, and a very low recombination percentage, that is 1.3%, that we have seen in the last class. And he suggested that the reason for the high percentage of parental combination is due to a phenomenon called a linkage. And he suggested that many genes can be located on the same chromosome. And all these genes located on the same chromosome are called linked genes. And the group of genes located on the same chromosome is the linkage group. And they tend to inherit together. So as they tend to inherit together, generation after generation, the parental combination always remains very high because they cannot be separated or assorted. So that is what we had seen in the last class. And the last class, we entered the last class with a question. If the genes are linked, situated on the same chromosome, and they cannot be assorted or separated, segregated, why should there be a small percentage, even though it is very small, 1.3% of recombination occur? Why does it happen? It should not happen actually. 100% parental combination should be there. So we concluded the last class with this question. And before going to the answer to this question, let us consider one more dihybrid cross conducted by Gallagher-Merrill, that is cross B. And in this cross, he selected again two characters. One character he selected was the color of the eye color. Two traits are there. White eye colored individuals are there and red eye colored individuals are there. This is the character, this is the same character that he selected for the cross A. And the second character he selected for the cross B is the size of the wings. Size of the wings. So as far as size of the wings is concerned, two traits are there. There are long-winged individuals and miniature-winged individuals. Miniature wing means short wing. So long-winged individuals are there. Miniature wing or short-winged individuals are also there. And interestingly, the gene for the size of the wing is also situated on the X chromosome. The cross B, so now we have three genes which are situated on the X chromosome. Size, body color, the gene for body color, gene for eye color, and gene for wing size. So these three genes are situated on the X chromosome. So now in the cross B for Morgan, consider only the size of the wing size and the color of the eye. So F1, he produced the F1. T.H. Morgan crossed a white-eyed female with a miniature wing, with a male having red eye and long wing. And here, the white eye color is recessive. We have already seen that. And the miniature wing is also recessive trait. And the red eye is the wild type, that is dominant. And the long wing size is also the dominant feature. So these two are the wild traits. That means the male is a wild variety. He crossed these two. White-eyed miniature wing female was crossed with the red-eyed long wing male. And he obtained the F1. And as in the case of cross A, here also, the F1 produced different types of individuals. Males and females were different in their genotypes, and phenotype also. F1 females were with the red eye and long wing, and males with the white eye and miniature wing. He crossed these two in the F1 as usual. The F1 female were crossed with the F1 male and produced the F2. And the F2, as in the case of cross A, he calculated the percentage of parental combination and recombination. Here, to his astonishment, he discovered he could calculate, he could find out an entirely different percentage, entirely different parental combination percentage and recombination percentage. The experiment was the same, only the characteristics were different. It was dihybrid cross and the procedure was same. But in the F2, he got a parental combination of 62.8% and a recombination of 37.2%. So this was drastically different from the percentage he got as a result of the cross A, even though the procedure he adopted was the same. Both were dihybrid crosses. Then he analyzed what could be the reason for this. What could be the reason for this difference in the percentage of parental combination and recombination between the F2 produced in cross A and cross B? And he suggested another phenomenon behind this. That is crossing over. There is crossing over. So we should remember that in the cross A, the percentage of parental combination percentage is 98.3% and recombination percentage 1.3%. And the cross B, the parental combination percentage is 68.8% and recombination percentage is 37.2%. And the reason for this difference, and also the reason for the occurrence of recombination, whatever may be the percentage, the reason for the occurrence of recombination, even though the genes are linked, is due to a phenomenon called crossing over. I know you had already studied crossing over when you studied cell division. So let us just briefly see what is crossing over. What is crossing over? Crossing over is a chromosomal behavior taking place during meiosis 1 of gametogenesis. So specifically, it is taking place in the packaging stage of the prophase 1. You know that. So during the zygoting stage, we have seen that the homologous pairs come together and exist in the tetrad form. Each chromosome in the bivalent form, and two homologous chromosomes putting together form a tetrad stage. So in the tetrad stage, the chromatids of the homologous chromosomes are called non-sister chromatids. The chromatids of the same chromosome are the sister chromatids, and the chromosomes of the other chromosome in the homologous pair is the non-sister chromatids. And in the packaging stage, as you had studied earlier, in the packaging stage, the non-sister chromatids of the homologous chromosomes cross each other, and the point where the crossover is called the chiasma. And after the crossing over, after they cross each other at the chiasma, the chromatids are broken at the chiasma and rejoin to exchange the chromatids, exchange the segments of non-sister chromatids. This you had already studied. Okay. So when crossing over and exchange of chromatid segments happen, naturally, the genes located on these segments also would be exchanged. So T.H. Morgan suggested that this exchange of chromosomal segments or chromatid segments between the non-sister chromatids of the homologous chromosomes is the reason for recombination. Is the reason for recombination, as you see in the picture here. Then the question here is, why should there be a difference in the percentage? So in the cross A and the cross B, when we examine, we can see a difference in the percentage of parental combination and recombination percentage. Recombination percentage is low in the cross A and it is high in the cross B. Why is it happening so? What is the reason for this difference in the recombination percentage? So let us examine the cross A once again. The cross A, in the cross A, two genes were considered. These two genes were located on the X chromosome, the gene for the body color and the gene for eye color. Note the position of these chromosomes in the diagram. You can see that on X chromosome, they are closely situated. The distance between these two genes is very less. They are closely situated. Okay. And the second cross, in the cross B, two genes were considered. They are also situated on the X chromosome, gene for eye color and gene for size of the wing. See the position of the X chromosome. They are situated apart. The distance between the gene for eye color and the gene for wing size is more than the distance between body color and eye color. So, is there any relationship between this distance and the recombination? So, T.H. Morgan made it very clear that recombination is due to crossing over. And he ascertained that the percentage of recombination is related to or is directly proportional to the distance between the genes. Let us see this. Suppose this is the chromosome, and this is the gene for body color, and this is the gene for eye color, and this is the gene for wing size of the individual. All these three genes are situated on the same chromosome. Now you look at the distance. The distance between the body color, the gene for body color, and the gene for eye color is very less. They are closely situated. So a chance for a crossing over to take place between these two genes, to separate these genes, is very less. That means chances of occurrence of crossing over between these two genes is less. So naturally, the percentage of recombination would also be less. Okay, recombination means they should be separated, and this gene, this part of the chromatid should be separated and joined with the non-sister chromatid of the homologous chromosome, where its dominant allele or recessive allele may be present. As it is depicted in this diagram here. And you see here, the gene for the eye color and the gene for the size of the wing here is situated apart. The distance is more. And the chance of crossing over between these two genes is very high. So more chances are there for crossing over between these two genes than between these two genes. So even though these two genes are linked, chances of getting separated through crossing over is more here between these two genes than between these two genes. So, in the second cross, where it's cross B, chances of the genes for eye color and body size getting separated is more. As a result, the percentage of recombination is also high. Whereas in the cross A, the chances of genes for body color and gene for eye color getting separated through crossing over and to form recombinant individuals is less. So that is why the percentage of recombination remains less in the cross A, and the percentage of recombination remains high in the cross B. I hope you understand. So beyond doubt, T.H. Morgan ascertained that percentage of recombination is proportional to or related with the distance between the genes in a linkage group. The closer the genes are, the lesser the chance for recombination. The wider the genes are, greater chance for crossing over to occur and for the formation of recombination. So based on this concept, one student of T.H. Morgan, Alfred Sturtevant, his name is Alfred Sturtevant, designed a technique of chromosome mapping. What is chromosome mapping? It is determining or assigning the position of genes on the chromosome. Relative position of genes on the chromosome. And depicting on a paper. Say chromosome can be represented as a single line, and on this line genes can be represented by dots, and the relative position of the different genes on the same chromosome can be noted by this method so that we can understand where the genes are located on the chromosome. What is the position of the genes on the chromosome? This is not an easy job. For doing this, hybridization experiments should be conducted, considering different gene pairs. Okay, if you want to know the distance between gene A and B, those organisms with these characters controlled by gene A and B should be crossed, and their F2 should be produced. The percentage of recombination should be noted. And for B and C, the distance between B and C, those animals with these traits controlled by gene B and C should be closed, their F2 produced, and the percentage of recombination should be calculated. Okay, so it should go like this. Doing this repeatedly for all the genes on the same chromosome, the distance can be calculated, and this distance can be plotted on a line, and that is very helpful to identify the location of a gene on the chromosome. So this is called chromosome mapping, and this was designed by a student of T.H. Morgan, Alfred Sturtevant. Okay, so by the experiments of T.H. Morgan and Drosophila or fruit fly, two concepts as far as the importance of two phenomena in genetics was revealed. That is linkage and crossing over. Okay? Now we shall wind up the class here. We'll see in the next class.